Guide to Silencing Genes by Quentin Main. The purpose for my lab research was to silence genes BLI1 and DPY11 in the C. elegans to alter the offspring phenotype of the wild-type parents. I performed this experiment to understand RNAi since it is such a new technique in the medical field, and I really just wanted an idea of what it was. I believe if I use the RNAi technique on wild-type L4 stage C. elegans, then the genes DP11 and BLI will be silenced, thus altering their phenotype. I chose the LE4 or L4 stage C. elegans because they are at the stage where they are about to produce offspring. So if I put in the E. coli strand OP50 early enough into it, they will produce offspring with altered phenotypes. So for procedures day 1, I inoculated the liquid bacteria with the E. coli strain OP50. I injected the OP50 into the liquid bacteria in hopes that I would be able to feed the new strain of bacteria I created with the E. coli to the wild-type C. elegans. I was doing this in hopes that the C. elegans would then eat the new bacteria without rejecting it, and this would alter their offspring's phenotype. So what I did was I incubated the tubes after injecting the OP50 to allow the OP50 to mix with the liquid bacteria as to put it on the NGM light plates. For procedures day 2, I used a bolt pipette to put drops of the liquid OP50 and liquid bacteria mix onto the NGM light plates. Then I grew the plates face up at room temperature to avoid the molding, and if it did mold, I would have to throw away the plate because the agar becomes contaminated and the C. elegans would not be able to survive on the mold. So the OP50 mix with the liquid culture then solidified and grew onto the plate, so when I do in the future put on the agar with C. elegans, they will be able to eat and then hopefully produce offspring with altered phenotypes. For my day 3 procedures, I finally began transferring C. elegans from the control plate to both the BLI1 and DPY11 plates. I then waited multiple days to allow for new offspring to be born from the L4, which would now be L5 C. elegans, that have already produced the offspring onto the new plates. I then waited for the offspring to grow into L4 C. elegans, which took about 3 days. This allowed enough time for the L4 C. elegans to show signs of gene silencing. In my further slides, I will show you the data I collected. So this was my first data chart I created for the DPY11. As you can see, it wasn't as successful as I hoped, but there was still a few C. elegans that showed signs of gene silencing. So I started off from day 1 to 0 and 1 to day 4, which I had 1s and 2s. Still a couple zeros, but that's okay as long as some data was shown. And on plates 6 and 7, the molds really took effect on the first day, which really hurt my results a lot, but I think that I still had very good results for my first time performing this experiment. So as I said before in my data slide, I had very slim results. I mean, I wish I could have had better results, but it was difficult at times. So as I said before, the days went on, and I had increased results from 1 to 2, which C. elegans with lack of gene expression, DPY11, showed more signs of the expression being silenced. So this data chart was my BLI1 expression gene, and as you can see, the results are much, much better than the DPY11. The first day was probably the best day I had, especially with plate 2. I don't know exactly why it showed so many C. elegans with the lack of gene expression, but it made me very happy to find out that I had very good results. And another great thing was there was no mold on any of the plates until after day 4, which was very good for my results as well. So as I said in the earlier slide with my BLI1 data, the BLI1 data showed much better results than the DPY11s, which was very good. The results per day were somewhat sporadic, and I really didn't have signs of increasing or decreasing, which is not necessarily good, but it's also not bad. So I'm very happy with the results I got from the BLI1 data. So my overall experiment with the RNAi technique was somewhat hard at first to understand what I was doing, but in the end I think I really have now a clear understanding of what RNAi is and how it functions. My experiment really helped me with the understanding, and it proved my hypothesis correct, which was really good for me. If I ever perform the experiment again, I would change some things, like maybe I would put the plates in a lighter or darker place, or even a colder or warmer place, because I would really like to see if my results would increase or decrease depending on the environment I place them in. So next time I perform this experiment, I will definitely have to try a few more things than just what I was given.